Co-immunoprecipitation and immunoblotting. To understand what co-immunoprecipitation is, we first need to know what immunoprecipitation is. Immunoprecipitation is a protein isolation method that uses specific antibodies that attach to the epitope of an antigen in order to precipitate the protein of interest via ultracentrifugation from a solution that contains other proteins. Co-immunoprecipitation is a process that precipitates an entire complex of proteins from a solution containing multiple and even thousands of proteins. An antibody attaches to the antigen of a known protein, therefore allowing all the other unknown proteins bound to that protein of interest to precipitate along with it. Immunoblotting, also known as Western blotting, is a technique that is used for the analysis of individual proteins in a protein mixture. It provides size and relative abundance in the sample. This technique is used to confirm tests for HIV, prion disease, hepatitis B, and herpes. It detects the level of protein expression in a cell. Co-immunoprecipitation uses antibodies which are attached to magnetic beads in order to easily precipitate the protein of interest. The antibodies then bind to the antigen of the protein of interest while all of the other proteins bound to that protein bind to the antibody as well. Once the solution is centrifuged, the protein complex precipitates and the rest remain in the supernatant. The pellet produced by the centrifugation can then be used to identify protein-protein interactions and study other members of the complex. There are generally 7 standard steps to immunoblotting. The first step is sample preparation. First, a sample of tissue or cells is broken down. Buffers are usually added to lyse the cells, and centrifugation is used to extract the protein. The sample is then loaded onto wells of SDS-PAGE, where they are separated based on charge and size. Smaller proteins move faster than larger proteins and move down to the positive pole of the gel as electric current is applied. Blotting. Once the proteins are separated into bands, they are transferred to a membrane carrier. A membrane is attached to the gel along with 2 buffer-soaked filter papers. An electric current is once again applied and the proteins are transferred to the membrane. The membrane is then rinsed and saturated with bovine serum albumin to prevent antibodies from binding to the membrane. This is also known as blocking. The blocked membrane is incubated with a primary antibody so it binds with the targeted protein. A secondary antibody is added so that it may bind to the primary antibody, thus creating a complex. The secondary antibody is enzyme labeled, and it may be alkaline phosphatase or HRP. The reaction mixture is incubated with specific substrates which give a visible color product that can be further analyzed with scanners, X-ray films, and chemiluminescence methods to quantitatively assess the protein of interest. Sensitive detection of fluorescence in Western blotting by merging images. So the key question for this experiment was, can the use of high fluorescence intensity dyes enhance detection sensitivity? Fluorescent microscopy was used to obtain high resolution images, since the traditional chemiluminescence method cannot be used to detect multiple proteins. High fluorescent intensity dyes were used to enhance the target signal. The image to the right shows joint images of the elution series of GST in the background area. An anti-GST antibody was used and fluorescence dye was conjugated to the second antibody. The different dyes on the left of the image have different intensities, which are the product of its molar extinction coefficient and fluorescent quantum yield. The different dyes show different intensities for 1.6 nanogram GST, which is proportional to the relative integrated density. Hence, the higher integrated density, the more clear the bands. And as you can see from the image, the more GST present, the more clear the band. According to the figure, the use of high fluorescent intensity dyes did enhance the detection sensitivity of GST. The pros for this experiment are that the use of different high fluorescent dyes in the experiment allows us to choose which dye we want to use for better and more clear images, which make this method suitable for quantitative protein expression analysis. One major drawback from this experiment is that the exposure time from the fluorescent microscope for the yellow, green, and red wavelength regions was different. So they weren't exposed to the fluorescent microscopy equally. They were exposed to light at different times. So one question I have for you guys is, how would the relative integrated densities and signal intensity be affected if the light exposure from the fluorescent microscope had been the same for all of the fluorescent dyes? These are my references.